My pleasure to introduce Dr. Jan Nolta. Dr. Nolta is a UC Davis professor and Department of Cell Biology and Human Anatomy at the School of Medicine. She'll be speaking on stem cells today. Dr. Nolta is the director of the stem cell program at the University of California, Davis School of Medicine in Sacramento and directs the Institute for Regenerative Cures. She also serves as the scientific director of the large UC Davis Good Manufacturing Practice Facility. And I could go on, but I think I will leave the screen to her. And thank you, Dr. Nolta, for joining us. We appreciate your participation.
Well, great. Thank you so much for that great introduction. And this society is very near and dear to my heart because I myself suffered from neuropathy from chemotherapy that I had 8 years ago, and I'm still stuck with it. So I just wanted to talk a little bit today about what stem cell therapies are, what's going on, what clinical trials are happening, what's going on in those clinics that you see advertised that might not be the real deal, and wanted to mention some of the things you should look out for. Are you seeing the slides okay? It's the slide view. We never know.
Yes, you're good.
Okay, good. Very good. Thank you. Okay. So I will just go forward, hopefully. It seems like it's different every time. There we go. So these are relatively new fields. Stem cells, you'll hear about immunotherapy for cancer patients, gene therapy, adding a copy of a gene to replace one that's broken, gene editing, doing that more precisely, and regenerative medicine. And these new therapies are changing the face of healthcare. These are living medicines. So the cells and the genes and the vectors that deliver the genes, they're actually alive, and we need to handle them in a specific way. It's not like a pill in a bottle or a vaccine on the shelf or in the freezer. It's a living entity. So we need specialized facilities, a high degree of teamwork and expertise to manufacture, formulate, and deliver these new living medicines. And we usually keep cells for delivery in cryopreservation under liquid nitrogen until we need them. And so I'm very, very lucky to get to direct the UC Davis Institute for Regenerative Cures. We're here in Sacramento at the medical school, the medical center. And we do a lot of training, and we also have a large facility to manufacture the therapies for clinical trials, FDA approved clinical trials to go to the patients. And this is what our manufacturing room looks like. So let's see if I can get me, there we go. So when we get cells, for instance, from a patient's bone marrow, we'll bring it into this clean room facility. We grow the cells in these incubators that have oxygen and are at the right temperature, mimicking your body. And we expand them in maybe in these this is a big bag where they'll be expanded, and we put in media and protein. And then they can be purified and made sure they're clean and delivered to the patient. And so this is something of which we're very proud as this facility. It started out as a big warehouse that used to be a part of the state fair, and now it's this high-tech facility. So we have this good manufacturing practice facility. And just saying up front, if you're going to get any kind of a cell therapy anywhere, you want to make sure that the cells have been manufactured or they're really expanded. We don't create them. We take them from the body, we change them in some way. I'll tell you about the different ways that we do that in a moment. You want to make sure they've been handled in this type of clean room facility. Otherwise, you could be, setting yourself up for infection or something worse, or the cells could attack you or form a tumor. So you really want to be very careful that people have followed the, we call them standard operating procedures, the protocols to handle and grow up and manufacture these products. And so we work with many academic and industry partners to make these therapies. We currently have, actually I didn't update this, we currently have around 40 stem cell and regenerative medicine clinical trials going, over 20 in the pipeline. We do work with peripheral artery disease, revascularization. We have a lot of neural disorder research going on, and other types that I will talk about today. Also a lot of sports medicine, where we may see patients with different neuropathies. The green arrows are those where we're doing clinical trials, and then the red, we're talking to the FDA to start. So there are different types of stem cells, and you'll hear mostly about these embryonic stem cells. They're the ones that have really grabbed all the headlines for the last couple decades. When people hear stem cell research, they immediately think of embryonic stem cells, but we don't actually use those because they were matched to one patient. So we have this thing called the immune system that would reject those cells if they were injected into a person. So we use a different type of pluripotent stem cell that can make any tissue. They can make nerves, they can make muscle, cells from your organs. We use something called induced pluripotent stem cell that we can make from the patient themselves, and that's a far more interesting field, and I'll talk about that in a moment. Most of the ongoing clinical trials and everything that you'll see in those clinics that are offering stem cell therapies for a high price are these somatic cells that are already in our bodies, and those will be harvested and expanded. So several different types of stem or progenitor cells, and I'm going to introduce you to each one. So I mentioned that we really don't use the embryonic stem cells. These are from in vitro fertilization clinics. They could form a whole human if they were implanted. They're a fertilized egg. If they were implanted into a surrogate mom rather than being thrown away, they could make a whole human. So they're very, very controversial. We don't use those. We're using the induced pluripotent stem cells. This is a colony of about 1,000 of these little tiny pluripotent stem cells, and they can really make any tissue. So it's very exciting. The way we do this, we take a little sample of the skin from a patient, and then we get the fibroblasts out of the skin. We grow these out, giving them sugar and protein and all the things that they need to live in a dish or a flask. We then add some genes that will basically open their chromatin and turn them back in time so that they're not differentiated and they're not a fibroblast anymore. They can become any tissue in your body. And this science is just remarkable that the field can even do this. And these then become induced pluripotent stem cells, and they're small colonies of small undifferentiated cells. We put them into flasks with certain growth factors and proteins and sugars and differentiate them along the way into the tissue that we need. We do characterization and sequencing, study disease in a dish from an individual patient. I'll give you an example in a moment, what we did with some neurons. And then we can differentiate them and theoretically transplant them back into the patients. Those clinical trials are just starting. They've started with some retina, transplant of retinal cells that were made in a dish like this. And some heart cells in Japan have been transplanted back into the patient. But this is a very new field, just about 15 years old, and not a lot of clinical trials with these pluripotent cells, but they have all the potential in the world. They can make, we work in our group with making tiny little human livers from them. We make brain organoids, pieces of eyes, all sorts of things. Here's some beautiful neurons that were generated from those cells from a Parkinson's patient. These were generated in the dish, and you can see that they have all of their, once they're differentiated, they have all of their connections. They signal to one another. We can chase the electrical signaling across the dish, put an electrical signaling in one, and it transfers across the dish to all of its neighbors, just like regular signaling that would happen in your brain, so or in your periphery. So it's really, really interesting to be able to make these cells from a specific patient with that patient's DNA, differentiate them into neurons, and then study them. We worked on, let's see if I can do this, not sure. Maybe not. Well, we worked on some patients that had Fragile X, and we made the induced pluripotent stem cells and then neurons from those patients. Oh, now it's not going to let me go forward, of course. There we go. And then we did everything to study them. We started out with fibroblasts from a female patient, made them into these induced pluripotent stem cell clones, did neuronal differentiation, studied everything from them, looked at synaptic proteins and density and calcium imaging, and made sure that they had normal chromosomes. Did all the staining to show that they're first pluripotent and then became neurons. And then it was very interesting. What we found out was that these neurons differentiated from the patient that had the disease had the same types of disorders that the patients would have. When we made them signal, they would start to get excited, and then they'd stay excited for a long time and then go back down. And the patients were suffering from different types of epilepsy. And so it was mimicking what the patients had. There's a lot of these types of studies ongoing. And so we have a team led by Dr. Kyle Fink here, who's using the induced pluripotent stem cells made from different patients who have Angelman syndrome, CDKL5, juvenile Huntington's disease. These are from kids. We can make their neurons. You can't go into the brain and biopsy what their neurons are doing, but we can make them in a dish and we can study them. And so it's really a lot of potential to have this. Wanted to talk about the blood forming or hematopoietic stem cells. This is the oldest stem cell therapy. They began in 1956, even a little bit before my time. That was with bone marrow transplant. By now, hundreds of thousands of lives have been saved. And we do use cord blood and stem cells mobilized into the peripheral blood rather than always going into the bone marrow to get these stem cells. So let's see, I started out working on bone marrow stem cell gene therapy with Don Cohn, who's one of the leaders in that field. We would take the bone marrow or cord blood when the baby was born, add the gene that was broken. These kids had bubble baby disease or adenosine deaminase deficiency. We knew from prenatal screening that there was, and this was 30 years ago when we started this, there was one gene that was broken that was ineffective, and that these kids would have absolutely no immune system due to that one enzyme that was broken, adenosine deaminase. We could use a viral shell, the shell from a virus, to deliver the DNA into the cell, and then do the bone marrow transplant back into the child with the engineered bone marrow. And we had some success, but we didn't get a lot of the cells marked back then. And this was published in 1995 with Don Cohn, who is the leader of really the leader of stem cell gene therapy. Go forward a few years, I went on to a different career. I worked with Don for 15 years, and then I went on to work with other types of stem cells, and Don kept at this now for 30 years, and now is really curing children. This is little Evie. She's one of the kids who has had her complete immune system restored by this type of bone marrow stem cell gene therapy. And that's Don, best mentor in the world. And Don just yesterday came out with this publication. It sounds very fancy, but autologous ex vivo lentiviral gene therapy for adenosine deaminase deficiency. This is putting the broken gene into the bone marrow of the kids and transfusing the bone marrow back into the children. You just put it in the vein and it goes back to the bones. And now over 50 children between his trial and one done by their partners in the UK, over 50 kids have been treated. And this is just a really beautiful curve showing that, I can't get this down anymore, but anyway, showing that they really had no events of infection or problems with the kids in 50 kids followed for up to 36 months. And so this type of stem cell therapy really is a cure, but it's very specialized. This is not what you're going to get in your corner clinic that's advertising, next to the 7-Eleven, come and get your stem cells. This is done in a hospital. The children are conditioned to receive their own bone marrow after it's been modified. You have to use some chemotherapeutic drugs to clear out some of their marrow so that you can get the corrected cells in. And so that's a type of bone marrow stem cell therapy that's really working and is curative, as are bone marrow transplants in many ways. But again, it's very specialized. So what's very interesting for us is that microglial cells in the brain come from those hematopoietic or blood-forming stem cells. And so the bone marrow transplant with gene therapy that I just described for you for immune system actually has a chance to impact some diseases that affect the brain. And so we are working with several of these diseases using the same techniques. A trial was done with hematopoietic stem cell gene therapy for cerebral adrenoleukodystrophy. This is a disease that mostly boys get, and it's a neurodegenerative disease. This trial showed that it was safe and that the boys did not progress further with their disease for several years. Right now, there are 87 clinical trials listed in the USA, and this is clinicaltrials.gov, which is where you can look for clinical trials that you might enroll for. You put in the term and where you want to look. But right now, 87 listed in the USA for hematopoietic stem cells and gene therapy. And our teams at UC Davis are working toward these HSC gene therapies for several diseases that affect the brain and the nervous system. Tay-Sachs disease, lysosomal storage disorders. These can kill children very young, Sanfilippo, Gaucher disease, other types of disease, hurlers. Of course, for leukemia and lymphoma, hemophilia, a clotting disorder, sickle cell disease, HIV to provide resistance against further viral insult. And then these that affect the brain, the CDKL5 disorders, Huntington's disease, juvenile Huntington's disease, Angelman syndrome, and PPP2 or 5D disorder, also called Jordan's syndrome, which is a little less of a mouthful. So then the last type of stem cell that I wanted to talk about, and I'll go into these in some detail because there are a lot of clinical trials ongoing with these. And if you're being offered a therapy at a clinic that's not an approved clinic, it is probably this type of stem cell to which they're referring. So I wanted to go into these in some detail. So these are mesenchymal or supporting cells. You can see them as the broad flat cells here lining the bone marrow. They protect and nourish the hematopoietic stem cells that I just discussed. And they can also be called nurse cells, and we also call them paramedics. They are reparative adult stem cells. You can get them from bone marrow. I hope nobody's having lunch right now, but you can also get them out of adipose tissue from liposuction surgery. And this is what a lot of those clinics do. They take out some liposuction, take out some fat, maybe from your tummy or somewhere, and then clean it up and reinfuse the cells. And so I'll talk about that in a few moments. We call them the paramedic cells because these are adult stem cells. They can't make an entire tissue like the induced pluripotent stem cells or embryonic stem cells I introduced at the beginning, but they can repair tissues. They respond to the scene of the injury inside our bodies. If there's a minor ischemic injury, or we get cut, or there's a deep bruise or a bone is broken, they will migrate to the scene of the damage, and they produce healing factors, and they can keep the tissue alive and get blood flow back to it. And that's really their job in the body. And so we call them the paramedics because they go to the scene of the crime. They do have a strong safety profile in clinical trials. And this is what they look like. I said they rush to the area. These red ones are the mesenchymal stem cells, and the green are some damaged muscle fibers that we've dissociated and put into a dish. And so you can see the red labeled mesenchymal stem cells crawling around and checking out the other cells. And there are these little red microparticles. They take stuff from the inside of themselves and they hand it off to other cells to try to keep the other cells alive. And so you can see, let's see right here, this cell has put a red piece of itself into the green damaged muscle fiber. And so we study this in pretty great detail, trying to understand what's passed from the mesenchymal stem cell into the damaged tissue. And one of the things that's really exciting is that these cells can actually transfer mitochondria, or the battery of the cells. That's what generates the energy inside your cells. The MSCs can actually transfer these into a damaged cell by what are called tunneling nanotubules. And it's like the stem cell just kind of opens up a gas hose into the other cell and starts pouring these new batteries into the other cell, along with factors and proteins to keep it alive and happy. And so they're really interesting cells. We do need to learn to harvest them in the right way, expand them in the right way, deliver them properly, and do the legitimate clinical trials. So these are, these are paramedics at the scene of an injury. They can really reduce the inflammation so that the tissue can heal. Unfortunately, they're pretty rare. They're 1 in a million cells in bone marrow, and just slightly more than that in fat in the adipose tissue. We need to isolate and expand them or grow them up to larger numbers in a clean room facility that I showed you at the beginning in order for them to be effective. And so adipose tissue is shown here. This is from a gastric bypass. We get just samples of fat. It also contains these mesenchymal stem cells, but it also contains many other cells. And so what these, these offshore or increasingly in the United States clinics do is they take some of this out through liposuction. They spin it down, they take all the oils off and stuff, and they get a cell layer, but there are a lot of other cell types and a lot of other stuff in there. They have injected this into people's eyes for blindness and made them more blind. They've injected them into people's bloodstream and caused clots and strokes. And so it can be extremely dangerous. There's stuff in there that you do not want injected into a wound or in your eye. You just want it to stay in your fat, or you want to diet it away. You don't want to move it to some other part of your body. And sadly, there are unregulated stem cell clinics that are doing this. And so the FDA has been going after them slowly but surely. The clinics have jumped on the bandwagon of the successful stem cell therapies that I showed you at the beginning because everybody's excited about those. There's unscrupulous people going after those of us who are desperate for cures for different things, for our kids, for ourselves, and advertising they can cure autism or Alzheimer's through injecting these cells. And these are largely cells from fat or taken from somewhere else in your body, but they're not growing them up in a clean room facility. They're just taking them and doing something to them and bringing them back and just injecting into you. And that's, the cheapest way for them to do it. They're not purified stem cells. Anything that you do when you take tissue out of the body, anything that you do that's more than minimal manipulation requires FDA approval and requires the FDA to look at a big pile of paperwork that you have showing that what you're going to put back into the human body is safe, clean, and effective. And these clinics don't have that, and they just kind of pop up and they make a lot of money off of people. They charge like $40,000 for these procedures, and then the FDA sends them the cease and desist letter, and they shut down and they move to a different place. So they're supposed to put up a sign that says not FDA approved. Definitely look for that. Here's one in Rancho Mirage. There have been a lot of different posts about these, but I just wanted to say a note of caution. There are a lot of them around California. We have some up in our area in Sacramento, as far north as Redding. They are definitely around. And so Beverly Hills has the highest concentration, interestingly. And I have to say, for cosmetic procedures like wrinkles and things like that, you might want fat injected. So if you have deep wrinkles in your face, they can take some fat from your tummy and put it in there with a little Juvederm or something to hold the cells in there. And you do actually want fat cells in there because they're plump and they're big and round and plump, and it can really plump up the tissue. But it's not growing the skin. It's not healing that skin. And that's the only case I can think of that these therapies might be doing something credible. So this is from my friend Paul Knopfler. He envisions a time when you're just driving up to get your stem cells. Pretty funny. And that's really all I was going to say about those clinics. When we have a question and answer, I'm happy to talk about, people's experiences or questions. I'm always happy to answer on emails. I do have a few more slides, but I'm always happy to answer questions about them on emails. But I did want to tell you, a couple of trials that are ongoing. Most of the calls that we get at our institution are for osteoarthritis. People have terrible problems with their knees and their hips as they age. And there are several clinical trials ongoing. The jury's still out. We don't know definitive results. Same for peripheral neuropathy. I haven't seen a clinical trial yet that is just like a landslide success. There are some ongoing that you can find on clinicaltrials.gov. But I wanted to talk about the one product, and by FDA approved, I mean it is in, it can be prescribed and insurance can cover it. So when you get through the phase 1, phase 2, and phase 3 clinical trials and it's safe and effective, then your doctor can prescribe it for you. But again, it's not just a product that you get off the shelf like a cement or something that would help your knee. It's actual real cells coming from juvenile cartilage. And this one is made of your own cells from the damaged knee. And so this is, if you've sustained some kind of defect in your knee, you've crushed it or had a athletic injury, you can see it here. They'll go in and they'll look at it. They'll go in and take out a biopsy around the damage, and then they grow the cells up in one of those clean room facilities. And this is all, governed by the FDA and following the standard operating procedures. And they regulate everything. They regulate the plastic that touches the cells. They regulate the type of serum that comes from cows that will go into the cell. They have to be USA herds, so we don't get any kind of spongy encephalitis or bovine, whatever that thing is. We have to monitor for every single virus out there to make sure there's nothing in the cells, and then it could go back into the patients. And clearly at the clinics, that's not happening, but for this product, it is. And so they will grow up the cells, customize it to the size, and then put it in. And they put it in with some fibrin glue, and it'll stick actually in the bone. And so that's kind of interesting for these. This is just the sheet. I'm happy to share these slides if anybody's interested, by the way. There's some links on there. And so that is the one orthopedic product that for knee damage, basically, that is FDA approved. I'm very lucky to work at the #1 vet school. And so we have canines and cats that come in with a lot of problems. And those are naturally occurring diseases of our companion animals, also horses that get athletic injuries. And so this is really a paradigm shift that we can take their cells or cells from another animal and test it on them for clinical trials for a certain breed of dog or a certain type of injury in a certain breed, like German Shepherds with osteoarthritis. And we can actually get enough animals that are either treated or standard of care that we can do these valid clinical trials. And so it's great to be able to do that. We have been doing spina bifida surgery. So by we, I mean the talented neurosurgeons that work at the UC Davis surgical, the Department of Surgery, and at the vet school surgical department. They've been treating bulldogs born with spina bifida. And this is where during development, the spinal cord is actually protruding from the discs in the back. It hasn't closed up properly. And while they're in the womb, the nerves are getting bumped around and damaged. The kids with spina bifida and the dogs are born crippled. They can't use anything. You can see where the surgery was done there. They can't use anything downstream from where that neural damage occurred. And most of these dogs will be put down at birth when it's found that they can't use their hind limbs. But there are these amazing bulldog rescues, and we have them here at our Shriners Hospital. Some people that work at Shriners rescue these little puppies, and our human and our veterinary neurosurgeons decided to start helping them. And they use stem cells on a little matrix to put in after they've closed up the defect. in the spinal cord. They use stem cells on a little matrix to kind of seal it up. They sew up the dura. They put the bones back in, sew up the skin, and they've now treated about 15 of the dogs. The dogs are walking and running around. They are still incontinent, interestingly. So the signals coming from the nerve saying that it's time to go to the bathroom or to alert someone to go to the bathroom, those are not intact, but they can stand and walk, which is really, really interesting. And so, yeah, this is Arthur. He's just one of the puppies that was treated, and they're also stinking cute. This has helped with the data, the safety data for a clinical trial that will be going on. Dr. Diana Farmer here at UC Davis will be treating babies diagnosed with spina bifida, and she'll be treating them while they're still in the womb, which is amazing to me. They do fetal surgery. They take the stem cells on the little patch. They do the surgery to put the nerves back into the spinal cord, put the patch on, refill the, I asked, what happens to all the fluid that the baby's floating in? They just put in saline. And they said that the mom refills it with all the electrolytes and everything after they sew it back up. They've done a clinical trial before, but not with the stem cells. And so this will be the first time that there's a fetal surgery using the mesenchymal stem cells on a patch, and those can put in the healing factors to help that spinal cord regenerate. And so we look forward to when, we can use these for coating maybe large conduits of nerves. We know that the MSCs can help with damaged nerves, with getting them the new batteries, the new mitochondria. They make nerve growth factor and all sorts of things, brain-derived neural growth factor, BDNF, all sorts of things that keep the neurons healthy. And so very excited about those clinical trials that will just get started. And those clinical trials for the humans are being funded by the California Institute for Regenerative Medicine. We're very lucky to have those there. So this is just the overview of everything that we do and what should be done before you get a stem cell therapy. There should be a lot of people working in labs with animal models, and then you get the FDA agrees to let you start the first clinical trial. There's an investigational new drug. That's all the paperwork that I told you about, the package that you send in. They then give you approval, clearance to start the phase 1. That's like 10 to 20 patients just for safety. Then you go into the phase 2, a controlled clinical trial. Phase 3 will be multi-center. A lot of people maybe at different universities or different hospitals. And then this gets to be an approved therapy reimbursed by insurance. And so with all those clinical trials, that whole pipeline of the phase 1, 2, 3, we have a alpha stem cell clinic team. And so for outpatient administration of these stem cells in regenerative medicine in the FDA approved clinical trials, and these are the stem cells that are grown in the clean room facility. That's an important distinction, following everything that the FDA recommends. And so the first thing that we did was for Duchenne's muscular dystrophy, and that has been a successful trial. This is a network of clinics throughout California, because it's funded by our state stem cell funding agency, CIRM. And so our goal is to deliver the high quality and safe stem cell clinical trials to patients. And these are the only cell and gene therapies that are approved for prescribing so far. So the field is pretty young. And the clinics are not delivering these, as certainly these are delivered in a hospital. So it's really a pretty short list. A couple of immunotherapies, using your body's own T lymphocytes to fight a cancer, but expanding them and adding a gene to them to target them to the cancer. More and more of these are in clinical trials. There's one for relapsed melanoma, lymphoma, leukemia. There's an approved gene therapy for blindness. I talked about the MACI for the damaged knee joints, and then the gene therapy for bubble baby disease, what I talked about with Don Cohn with the bone marrow transplant, approved in Europe and hopefully soon in the United States. And so it just really takes a village to think up, test, make, formulate, and deliver these cells over to the clinic. Our Alpha Clinic has what's called a STEM mobile to take them from our manufacturing center here in the Institute over to the patients in the clinic. And this is our Monday morning meeting every Monday morning. It's virtual right now, but we'll be back together pretty soon. So we thank CERM, who funds a lot of our program. Also, private donors will fund different diseases on which we work, and some from the National Institutes of Health. And that's me. I like to tweet. I use it for good, not evil. And we're we're here if you have questions. I always welcome emails, and I think I have some time to answer some questions. I raced through that pretty fast. So. Any questions?
Well, first off, thank you very much, Dr. Nolta, for a very good presentation and a good overview of what's available and what we should be careful and cautious of. So we want to open up the chat to some questions. So I have one here that says, of course, one of them comment was Arthur is amazing, which was really cute and very true.
So cute.
Yeah. So here's one question that says, can you work on patients that have had back, neck, leg, foot, shoulder surgery to help with pain?
That is a really good question. We actually have and I didn't mention it here because it's just not very close to the clinic yet, unfortunately, but we have a collaboration with Bruce Hammock here, who works on some interesting molecules that deaden the nerves to pain. They're eicosanoids. I don't know the chemistry of them, but we're engineering our mesenchymal stem cells to deliver those. And since they go to the areas of damage, right now this is in immune deficient mice where we can transplant in the human cells. So we're not even in the clinical trials. But you would need to have the cellular therapy delivering something to deaden that pain locally in the area where it's happening. The mesenchymal stem cells can reduce a lot of inflammation. So if it's inflammation causing pressure on the nerve, they might be able to do that. Regrowing the nerves is always a goal. A lot of people are working on that. So that's what we hope in the future. Now, back pain is different. We and others have, the spine center, and they can do a lot of different things, including stem cell injection into the vertebra to try to cause more cushioning and to try to get you more relief if the nerves are being pinched there. But that's a different type of pain.
Mm-hmm. Well, interesting that we also, the Western Neuropathy Association also worked with Dr. Hammock on some of his new medication that he's working, they're in clinical trials right now.
Yes,
We're
yes.
very anxious to have that completed so that where our hope is that it will help with a lot of the pain that we all suffer, because it's very excruciating and very, constant.
Yes.
Okay, there's another question that came here, and it says, are there stem cell possibilities in the pipeline for peripheral neuropathy?
There are, and they're in clinical trials. I really should have tried to dig into those more deeply. I'm not sure, what phase of clinical trials they're in, and that would be, delivering the cells locally for the anti-inflammatory effect that they can have. And the MSCs really are better than, steroids in many cases. They can do more than the steroids, but they don't stick around as long as we'd like them to, these mesenchymal stem cells. So we're working out many different aspects of that. So definitely people are working on it. People are thinking about it. It's just, what's going to be best and what's going to work best.
Mm-hmm. Mm-hmm. Okay, another question says, I saved my son's embryonic stem cells. They are now 11 and 13. Do you recommend I still keep them either for help with neuropathy or something else?
So probably umbilical cord when they were born. Was it the cord blood?
It doesn't say, but I'm going to, it could be.
Yeah.
Yes. Yes.
Yeah.
Yes.
So, so that will, those cord blood cells will match them. And, you know, if it's not a lot of money to keep them, I sure would, because we don't know what we'll be able to use them for in the future. And we can also use those to make the induced pluripotent stem cells. I didn't mention that. It doesn't have to be the fibroblasts. But think about, you know, your arm goes through all the damage from what you eat, and if anybody smokes, or there's, you know, toxins in the air or from the sun damage, those cord blood cells are the most pure cells that those sons will ever have, and they're banked, and so they might be needed in the future. I would hang on to them personally. I wish I had some. You know, they're completely unadulterated by anything that's happened, and they last. We know they're fine for at least 30 years in the freezer.
Now you actually have a cord blood bank, do you not?
We partner with cord blood banks. Yeah, I run the our program, not me personally, but the wonderful people who work with me, run the California Umbilical Cord Blood Collection Program. And we partner with different hospitals to collect the cord blood instead of throwing it away, and then put it into the public banks. So the bank isn't right here. It's actually in San Diego. We ship the cord down there.
Okay. Okay. Any other questions? So we'll give it just a few minutes here and see if they get any more here. I don't see too much right now, so
Yeah.
we'll. I think what we'll do is unless something pops up, I'll want to say again, thank you very, very much for your time and your expertise and for sharing information with us. I hope that it helps some of us to understand it a little better, and also for us to be a little more careful in terms of how we use some of the things that are out there. Here's a couple. Got a bunch more now.
Yeah, no worries.
Okay, so let's
Yeah,
go on with a
thanks.
couple more if you don't mind.
Sure.
Okay, so
Yeah.
here's one that says, is it impossible that the Wharton jelly in the umbilical can be a better solution?
That's a great question. And that's one of the places that we get the mesenchymal stem cells. And so I didn't mention that. I didn't mention all the different places that we can get them, but yeah, that's a very young source of cells, and they grow. They have very long telomeres, and they just keep dividing. So that's an excellent source.
Okay.
And for, you know, for the question about the clinical trials, although it's not my, you know, particular thing that I work on, so I'm not really as up on it as I should be, I would suggest going to clinicaltrials.gov and looking for legitimate places that have, you know, affiliated with a medical center or a university that are running clinical trials. And you can put in, you know, peripheral neuropathy and stem cells or gene therapy or whatever you want, and find maybe one in your region to try.
Okay, thank you. Sonia, I thought you were showing up your hand. Did you have something you want to ask? Because I don't see it in the chat.
Maybe it was a clap.
Oh, maybe that's what it was. Okay.
I do.
Oh, she
I
does.
do. You hear me?
Yes.
Okay. You know, now that we have the ability to check people's genes, look at their DNA to see if they have acquired any of the hereditary acquired neuropathies. Are you looking at ways for people who have these genes, the hereditary genes for neuropathy, to try and do things proactively for them to help minimize the damage from the hereditary disease?
Yeah, that's an excellent question, and other teams are. It's not my particular specialty, but anytime that we can pinpoint a gene that's going to cause trouble downstream, there can theoretically be a gene therapy developed to stop the effects of that gene. And so what another thing I didn't really have time to get into because it's a gene therapy talk rather than a stem cell talk, but there's a adeno-associated vector, and it's a very small little virus, and it's used to carry many types of genes, and it's injected into the bloodstream, and it's being used for all sorts of diseases now, hemophilia. And so I would imagine that would be the best one for this, and maybe for delivering some of the things that might combat the pain, because that can be injected locally. It just one time infects your cells. It doesn't, course through your body and keep, like HIV or something. It doesn't keep replicating. It's just a one-way delivery. And so there are people working on different modulators for different specific diseases. And this field is moving really fast.
Okay, thank you.
Okay, thank you for your talk today.
Good luck. Yeah.
And thank you.
Thank you.
I think that's probably where